My name is Arun Padmanabhan, and I'm excited to tell you about my work in the lab. I'm a physician scientist at UCSF, where my clinical practice is in general cardiology, and my work in the laboratory focuses on aspects of cardiac development and disease. As a graduate student, I studied developmental biology, where I really fell in love with the beauty and complexity of how the heart forms. As a medical student and resident, I was drawn to cardiology because of the wealth of data that drives clinical decision-making. I'm now using my unique background and interests as an adult cardiologist and developmental biologist to answer a series of questions with relevance to human disease. I'm studying the fundamental mechanisms that underlie how cardiac muscle cells develop and how they maintain their cellular state in the setting of stress. Now, these questions have broad relevance to why some babies are born with heart defects and also to what goes awry during heart failure. Now, the heart is a unique organ in that it begins beating in early gestation and continues beating throughout our entire lives. You can think about the heart like a fancy sports car. Everything about it has been finely tuned to maximize performance. And like a fancy sports car, when something breaks, it's not easy to fix. Unlike skin or liver cells, heart muscle cells can't meaningfully regenerate after they undergo some kind of insult, whether it be acute, like what happens in a heart attack, or chronic, like what happens in long-standing, untreated high blood pressure. It also turns out that the genetic programs that heart muscle cells turn on in the setting of stress are very similar to the same programs that govern early development. I'm interested in understanding the factors that influence how these genetic programs are modulated, where I'm focusing on a family of proteins that control how genes are turned on in these settings. Now, this has clinical relevance for two reasons. First, congenital heart abnormalities are the most common structural birth defect in babies born in the US. Second, these studies will have importance for the emerging area of regenerative medicine therapies, which are particularly relevant to a growing epidemic of heart failure, a disease with a really dismal prognosis. About 40% of patients will die within 5 years of being diagnosed with heart failure. This is worse than a lot of cancers. It's one of the most common conditions I treat in clinic and occurs when a weakened heart muscle can't pump out enough blood to the body. Now, things that used to seem like science fiction, like growing heart muscle cells in a dish, assembling them into biopatches to improve heart muscle function, they don't really seem that far-fetched anymore. And while they're far from prime time, all of these therapies will fundamentally require a foundational understanding of how to make a heart muscle cell. So I have elected to study a protein called bromodomain-containing protein 4, or BRD4, in cardiac development. And I've chosen to do so because this protein is known to be a critical regulator of maintaining a cell's identity. For example, when a skin cell divides, which is happening all the time in all of us, how does it know to remain a skin cell? Well, it turns out that skin cells, like all cells in the body, place little molecular bookmarks in the genome, like Post-it notes that say, hey, I am a skin cell. I need to remember to do the things that skin does. BRD4 and proteins of its class are responsible for reading these Post-it notes to maintain a cell's identity. We also know that in congenital heart disease, these Post-it notes can be misplaced or missing entirely. One tool that has been very powerful in helping us understand how BRD4 works is a drug that inhibits its ability to read these Post-it notes. I've used this drug, which is really a tool compound and not something that could be used in humans, to ask whether it could make a sick heart muscle cell in a failing mouse heart forget that it's sick. And these studies have shown some promising results. But more interesting to me is that we don't know the normal function of these proteins during development. And that's what I've been interested in trying to address. I hypothesize that BRD4 plays a critical role in normal cardiac development, and my work over the last year supports that notion. I've been taking a multi-pronged approach where I've been using both stem cells and animal models that I've created to explore the role of BRD4 in driving cardiac development. Now, a very powerful way to understand what a protein is doing is to either inhibit its activity with a drug or completely remove it genetically in a relevant model system. A great model system for understanding aspects of cardiac development is with stem cells in a dish, where you can take a defined cocktail of factors to make those cells turn into cardiac myocytes, or heart muscle cells. When I do that with mouse or human stem cells and add this drug that inhibits the activity of BRD4, I see fewer beating cardiomyocytes, consistent with a role for BRD4 in this process. I've also generated a novel mouse model where I can specifically delete BRD4 only in the cells that are going to form cardiac myocytes. And when I do that, these mouse embryos die during mid-gestation with gross cardiovascular abnormalities, again consistent with the critical role for BRD4 in this process. I've gone back and looked at unpublished data from patients with congenital heart disease, and I've also found a number of mutations in BRD4 in those patients as well, all consistent with BRD4 playing this critical role. I'm now turning my attention to mechanistically understanding how BRD4 is driving the genetic programs that make heart muscle cells. I'm using really powerful techniques that allow me to detect exactly where in the genome BRD4 is bound and all of the genes that are being expressed by single cells to get at this question. Taken together, I'm developing a blueprint for the circuitry within these cells that governs how BRD4 makes a cardiac myocyte. Ultimately, these studies will give us an understanding of how to make a heart muscle cell, which has relevance to understanding why some babies have heart defects and potentially how to even prevent them, and ultimately to improve therapies for patients with heart failure.